Okay, so we wrote on this diagram yesterday, and this was a little new, that this part we already knew that B cells become activated and divide and differentiate into memory cells and plasma cells that will secrete antibodies. The part that was new was the relationship between the helper T cells activating this and how helper T cells activate this process. So the B cells, we learned, can take in the pathogen and display its antigen, which then helps the activated helper T cells to bind to it. So in conjunction with binding, the helper T cell binding to the B cell, as well as secreting cytokines, stimulates the dividing and differentiation of these, and so then it secretes the antibodies. So the last little bit of this section is about what do antibodies actually do to help with getting rid of your antigen. And so that we haven't really talked about here. So antibody-mediated disposal of antigens. So antibody-mediated means that antibodies are going to help with this, and disposal means get rid of. So the binding of antibodies to these antigens is also the basis of antigen disposal mechanisms. So it's going to help to get rid of these antigens. Microbes are eliminated by phagocytosis and complement-mediated lysis. So that's what your next picture shows, is these two processes. So with this picture here, you have the binding of the antibodies to whatever pathogen, and there's different ways in this that this can happen. So the first box here says viral neutralization. Notice here, here's your virus, and the antibodies are binding to the antigens on the surface of the virus. And so what that does, how viruses work, is they actually bind to your cell membrane and then inject its genetic material into your cells and then cause your cells to become virus reproducing factories. The DNA, the viral DNA kind of takes over your cells and makes your cells make new viruses, which then can go out and affect other cells. So by these antibodies binding on the outside of the virus, these antibodies kind of are physical blockers, so the virus can't actually butt up to your cells and therefore can't affect them. So that's what that virus neutralization does. And then they do the same thing with the bacterium. And in both of these cases, when the pathogen is surrounded by antibodies, notice here it says it enhances phagocytosis by the macrophages. So the macrophages then and enhances by the actual antibodies can bind to receptors on the macrophage, and therefore causes this to happen more. And so therefore you get rid of the pathogen. And so that's the first part here that enhances phagocytosis. These two are similar. The only difference is this is dissolved in your liquid in your cell or in your body, and this is not. So agglutination, what does that mean? I want to show the bacteria. When the antibodies bind to the bacteria, remember antibodies are Y-shaped. So you have two binding sites that can bind to two different antigens. So what happens is, if you have a bacteria, the antibody can bind to the antigen on this bacteria, but then that same antibody can bind to the same antigen on a different bacteria. And then another antibody can bind, this is the other bacteria, antibody can bind to this bacteria as well as this bacteria, and so on and so forth. So you can get, so then you might have this antibody binding like this. And so you get this clumping of the bacteria, and then what happens is that enhances phagocytosis, and then you can bring in lots of bacteria at once and get rid of it that way. So that's the agglutination. Precipitation is kind of the same idea, except you have your antigens are soluble molecules, which means that they've dissolved in water. And so what happens is that when they're dissolved in water in these antigens, so there can be chemicals, because sometimes an antigen is not a cell or a virus, it can be a chemical, like a toxin that's released by a bacteria or something like that. So they can be dissolved in the water, and so these antigens can bind the antibodies can bind to these antigens, and when they group them together and clump together, they can precipitate, meaning that they form a solid in the water or the solid in your fluids in your body, and then that aids in the macrophages being able to take them up that way. So the moral of the story here is that the binding of the antigens enhances to the pathogens, enhances phagocytosis, which then helps to get rid of the antigen. So that's those two. Then the second thing here says activation of complement system. The complement system is a series of proteins. And so here's your foreign cell. So here's your foreign cell. This is the pathogen. And notice that our antibodies are attached to this. So it doesn't say this here, but these right here are your antibodies. And they would be attached to the antigens, which are kind of like little red parts here on there. So they're attached to the antigens on the surface. And so sometimes what this will trigger is proteins that our cells make called complement proteins that bind to the antibodies. What that triggers, it activates the proteins, and basically through a series of steps, the proteins cause pores to be formed, so it kind of pokes holes in the foreign cell. And what happens is then ions can go into the cell, the foreign cell. Ions are going to decrease the water concentration when they go in here, so that means that water will follow, and therefore the foreign cell, the pathogen, will swell up and lyse and explode, and therefore kills the pathogen. So that's another thing that antibodies do. So either way, most antibodies work this way, all right, and you get rid of them through phagocytosis, but some work this way, causing cell lysis as well. And so that's the importance of antibodies and what they do. Okay, so right now I'm gonna have you guys. We're gonna maybe work. This is not in your note packet. I'm gonna have you think about what we've talked about with the cells involved in the immune system, and we're going to relate it back to Chapter 11. So this says, show how the following interact in a positive feedback loop. So macrophages and dendritic cells, helper T cells, and B cells. So positive feedback loop. Anybody remember what a positive feedback loop is?
It helps amplify the signal and it just keeps it going.
That's right. So the stimulus, it keeps it going. So the stimulus causes a response. That response amplifies the original stimulus. So that one packet we did talked about it being a vicious cycle. All right, so it keeps on increasing. And so the example we used is with contractions in childbirth. And so the pressure on the cervix causes the release of oxytocin, which causes the uterus to contract more. The uterus contracting more increases the pressure, which causes more oxytocin to be released, and you get this cycle until, of course, what happens? The child is born, right? And so, so we want to figure out how this positive feedback loop relates to these cells. So in order to do this, you have to think about macrophages and dendritic cells. These are two types of antigen-presenting cells. So what do they do? And then how does that relate to what helper T cells do? And how does that relate to what B cells do? And how does that relate back to the beginning to make it a loop? Okay, so I'm going to give you a little bit of time to work this out. And you can work with each other if you'd like. And I'm going to pause. So we want to see how these things create a positive feedback loop. So let's start with the first bullet where it talked about macrophages and the macrophages and dendritic cells, which together are types of antigen presenting cells. And so what could we say about those?
It presents a pathogen fragment which makes a helper T cell bind to it, and then both the APC and the helper T cell release cytokines.
Okay, yeah. So let's start here with what you said with I'm going to put just the macrophages and dendritic cells. So you said that they display pathogen fragment, I think is what you said.
Yeah.
Okay. And so in order to do that, what do they have to do?
They have to use the molecule that takes it
HC.
MHC, yeah.
Okay, yeah. And in order to display that fragment that was once part of the pathogen, they have to take it in, right? They have to engulf the bacteria or pathogen, whatever it is, virus. So they engulf the pathogen and destroy it, right? And then, as Nathan said, that they display the pathogen or the antigen fragment from the pathogen. And said one other thing that they do, they release,
Cytokine.
yes, so we have all that going on here. What happens next? You said this affects another type of cell. What type of cell?
Helper T cell.
The helper T cell. So the helper T cell what does it do to this? It binds to the antigen, right? The fragment. And what I mean by that is the antigen fragment that is being displayed here. And then what's the other thing that's going to help activate the helper T cell? These cytokines here, right, that are being released. So they bind to the antigen fragment. I'm going to put here and receive the cytokines, those signals. And what that's going to do is now we have an active helper T cell. Let me get her test real quick. Active helper T cell, what's it going to do when it's active? You need to come up. Yeah, I'm going to put you next to him. When the helper T cell is active, it does two things. What is it going to do? Yeah, so this, the active, releases its own chemical signals, the cytokines. And what else does it do? It's active and it goes and is going to help activate another molecule or another cell. A B cell, right? So it's going to bind, all right? And binds to the B cell. So here we have now your B cell. And so let me show you here just in a little picture here. So here's your macrophage. It has taken in a pathogen, destroyed it, and on the MHC proteins, it displays the antigen. So this is your, I'll just put APC or antigen presenting cell. So here's your helper T cell, comes in here and has a receptor on it that's complementary to the antigen fragment. So there's your helper T cell here. And so, so this helper T cell becomes active by this binding and also the release of the cytokines from the APC, and so therefore, and remember, binds to specific receptors and activates the helper T cell. So now we have an activated helper T cell. Is going to come and it's going to release its own cytokines and bind to the B cell. How does it bind to the B cell? What needs to be true about the B cell? This we learned yesterday, that the B cell does something with the pathogen. Katie?
It presents part of the pathogen that,
Yeah, it presents part of the pathogen. So the B cell here, B cells are activated by. And the first thing is that the helper T cell binds to it, and this is where that the presentation of the pathogen comes into play. So if we this is your B cell right here. The B cell, remember, has those Y-shaped antigen receptors that are very almost identical to the shape of antibodies. So they have binding sites at the ends of those that the antigen can bind to. And then they take that in and destroy it, but you have these antigen fragments inside here. And then the B cell displays those antigen fragments. And so that's what we have here. That's how the helper T cell binds to it. So now this, going from this here, the active helper T cell can now go and bind with this. So this is your active helper T. Binding with the B cell, and so it's activated, the B cell becomes activated by the binding of the helper T cell. But what's the second thing that helps activate it? What does the helper T cell do? Yes, releases more cytokines, which activate it, all right? So the cytokines from the helper T cell. B cells activated. What happens when they become activated?
They make clones of themselves.
They make clones of themselves and then eventually differentiate into what two types of cells?
Memory cells and
Memory
plasma.
and
Plasma.
plasma cells that are going to secrete antibodies. Which one of those, memory or plasma cells, are important for your current infection?
Plasma cells.
The plasma. The memory cells are going to be important for later on, for the next time you get infected, but the plasma cells are important here. So I'm going to put here, I'm going to do 2 arrows here just to show that. So the B cells divide and differentiate. And when they divide and differentiate, that means they specialize. So the memory cells, I have the memory cells coming out of the loop because the memory cells aren't really involved in here, but that's what an activated B cell does. But it's your plasma cells, this part here, that are important because plasma cells are going to secrete what?
Antibodies.
Plasma bodies, these are going to secrete antibodies. Now, from this, how do antibodies, remember this is a positive feedback loop, so how does this activated B cell that's secreting antibodies enhance or amplify the beginning of our, so how do we finish that loop?
The antibodies help kind of catch the bacteria, they bind to the outside of it, and that makes it easier for macrophages to go through phagocytosis.
Absolutely. So that is back to this picture here that we just looked at. Remember when the antibodies bind to the pathogen, it aids in phagocytosis. So it allows phagocytosis to happen more. So therefore, the more antibodies that are secreted, it's going to activate phagocytosis that occurs more. And phagocytosis, so if we put an arrow here, these antibodies are going to stimulate more macrophages to engulf the pathogen, which means they're going to splay it more and activate more helper T cells, which will activate more B cells, will secrete more antibodies, and we continue going on and on and on until what stops our vicious cycle of this positive feedback loop?
The antigen is gone.
The antigen is gone, right? So you rid your body of the antigen, just like when the contractions with the baby's gone, we stop, all right? And so it doesn't continue forever. So positive feedback loops do have an end. How'd you guys do on that? Okay. Get some of it? All right. Okay. All right, so where we're at right now is what we've done so far is this is basically the end of how the immune system works, okay? The rest of the notes, we're going to talk about what happens when we receive other cells. So we're going to talk about blood transfusions, tissue transplants and how that relates to this. And then we're going to look at when something goes wrong with your immune system, so something disrupts it. That could be a disease, all right, and so on and so forth. So that's kind of the rest of the chapter, all right? So as we stand right now, the function of the immune system we've got covered. All right, so active and passive immunization. So in this class, we've talked a little bit about vaccinations. Active immunity develops naturally in response to an infection. Yeah, on this page. Can also develop following immunization, which is also called vaccination. And so as we talked about before, in an immunization or during a vaccination, you get a non-pathogenic form of the microbe or part of the microbe that elicits an immune response, and eventually we'll get memory cells that will then protect us when we come into contact with that pathogen later on in life. So we talked about it's the antigens, the shapes on the surface that are important. So when you're given a vaccination, you're given either dead or weakened or parts of the pathogen, but they've made it non-pathogenic, so you don't get sick. But the shapes of the epitopes or the antigens are different than your body's, and so your body launches a primary immune response against it, but it doesn't make you sick because it's a dead version of it, a non-pathogenic form. But the key is that you go through this immune response, and during your primary immune response, you make memory cells. So now you have these memory cells, and so then the next time you come into contact with the bacteria or virus or whatever in your everyday life that is harmful, then you launch an attack very quick, because that would be your secondary immune response. And remember when we looked at that graph, the secondary immune response is much faster and much more enhanced. You produce a lot more antibodies and so on, and so then you don't get sick as a result of coming into contact with that same bacteria or viruses. So we have gotten rid of basically, or really greatly reduced, a lot of very harmful diseases, or used to be deadly diseases where a lot of people died from them way back in the day, that people don't die of and we don't see very many cases of because of the vaccinations.
You know how they're like the often to take it like nasally, like for vaccination you get the office. Is there a difference in strength or how one affects you?
You know what, the nasal one, I don't know exactly what's in it. It doesn't, no, you still have the same type of response. You still have that primary immune response to that. So I don't think that like probably one is better than the other. In terms of, we talked a little bit about side effects and stuff of this in another class period here. So like that I can't speak to very much. But you can have side effects to things other than the actual weakened non-pathogenic form, because it's in a liquid, so there's other things in it. I know when I had my kids and me vaccinated for the flu shot this year, they asked me if my kids have ever had an adverse reaction to the shot before, because some people do. And then they asked me if they had ever had a reaction to eggs. So clearly there's some kind of something that's found in eggs, so for people who are allergic to eggs, like they would have an adverse reaction to. So there are other things in there that you can have an adverse reaction. That's hard to predict though, I guess. So clearly they've had enough people with it have a reaction to this, whatever molecule it was that's also found in eggs, right, for that.
I think the nasal thing is more of like a comfort. So like.
Yeah, but the effect is the same.
Needles and
Yeah,
stuff.
the effect is the same. I know my kids, I waited too long and they ran out of the nasal one. They were so mad at me because I didn't get the shot. My kids like to have the nasal one so that they don't have to have the shot. But apparently that's really cooler. And apparently the doctor told me that if you don't use all the nasal ones, you can't return them. Apparently you can order shots, and if you don't use them, you can return them and get kind of your money back, but the nasal ones you can't, so then the doctors don't order that many because they don't want to be out, like, from so that's a little side, right? All right, so that's so vaccinations are what we call active immunity. There's also something called passive immunity. This is immediate short-term protection. It's conferred naturally when it says IgG. I'm going to have here, this is a type of antibody. You guys don't need to know these antibody classes. This is also an antibody, another type. Conferred naturally when this antibody crosses the placenta from mother to fetus, or when another antibody passes from mother to infant in breast milk. And you can also get injected with antibodies. So let me talk about this. This is called passive immunity because you're receiving antibodies from somewhere else. The antibodies, when you receive antibodies, you don't make them yourself. So therefore you have these antibodies in your system for a small period of time, which means that if you're infected with the bacteria that antibody binds to, you have temporary immunity against that. So the antibodies will behave just as if you produced them, but you didn't. So this type of immunity is different than active immunity, where the vaccination, you're given the actual antigen, and your body actually has a full-blown primary immune response against it, and you have memory cells, and therefore protects you later on. With passive immunity, you never have an immune response. So, like for instance, when babies are in the mother, the mother's certain types of antibodies, there's certain antibodies that can cross the placenta and go from mother's bloodstream to babies. And so the mother's antibodies can go into the baby's bloodstream and provide temporary protection for the baby against certain bacteria and viruses. But the baby doesn't have long-term effects or long-term protection because they never had an immune response to that bacteria or virus. So when that baby is born and doesn't have the mother's antibodies anymore, and they come into contact with that bacteria or virus that the antibody was protecting, now that they don't have it, then they will launch a primary immune response. Then they'll have memory cells for the next time that they come into contact with. And this also, some antibodies are passed from mother's breast milk to babies. And so this is one of the reasons why breastfeeding, a benefit of breastfeeding is that you have this infant that has a very immature immune system because this infant hasn't come into contact with very many bacteria and viruses. And so they don't have memory cells really, so they can get sick really easily. And so these antibodies that are passed from mother to baby can provide temporary protection against certain, whatever those antibodies that protect, that the mother has against certain bacteria and viruses for the baby. And then this last one is injecting antibodies. So people who go, let's say, you're going to go on a trip to a third world country where a certain disease is prevalent that you may come into contact with. What they'll do is sometimes give you an injection of antibodies against that particular bacteria that causes that disease. So now you have antibodies floating around in your bloodstream so that now when you're in that country and you come into contact with that bacteria, the antibodies immediately bind to it, and therefore you never get sick because of that. But it's temporary because you don't have, because the antibodies are present and they take care of it and bind to the bacteria right away, you never launch a primary immune response to it. All right, so it's as if your body never came into contact with it. All right, so that's passive immunity. Oh, in this I put this picture here. I forgot I put this picture here. That just shows, because when I was talking about placenta and the mother and the blood and so on, I realized that you might not know what I'm talking about. So here's the baby, and here's the umbilical cord, and this is the placenta. And so this enlarges the umbilical cord, which has arteries and veins. And so the baby's blood flows through the umbilical cord, and this part right here is the placenta. So here's the umbilical cord connected to the placenta, and the baby's blood loops through and goes back. So therefore it comes out here, goes through these loops, comes back, and the purpose of that is this right here are veins and arteries of the mother's blood, and diffusion can occur between baby and mother's blood there. And so some antibodies can move across there and then provide temporary protection for the baby. So that's what that is about. All right. So this last little bit, we're going to look at what happens when we get cells from another individual. So we're gonna look at blood transfusions, transplanted tissues. All right, so let's look at blood groups first. So to understand this, we need to understand the blood groups. And so antigens on red blood cells determine whether a person has type A, B, AB, or O blood. Antibodies to non-self blood types exist in the body. And so transfusion with incompatible blood leads to destruction of these transfused cells. So what does this mean? So the antigens on the red blood cells, so we have these different types of blood. So A blood, why we say that they have A blood is on the red blood cells, so if this is a red blood cell here, you have antigens that we call A antigens. So I'm just going to draw them as little triangles here. So this is an A antigen. So people with type B blood don't have A antigens, they have B antigens, which has a different shape. So therefore, I'll just draw little semicircles here. So this is a B antigen. People with AB blood, anybody know, I'm a hypothesis, what they have?
Oh, neither.
Both.
Okay.
Both. You gotta shut it out.
You threw me off.
I did, I gave you a look. So they have both A and B antigens, and type O blood people, they have neither. So they have neither A nor B antigens. So what happens is, if you are a person with A blood, what you recognize as part of yourself is A antigens. So therefore, could you put B blood into A blood people? No, they recognize it as foreign because they don't have the A antigens, they have this B antigens. So people with type A blood have antibodies against B blood. All right, so people with A blood, so you can't put type B blood into a type A blood person. There's antibodies that will bind to it and then clumps the blood, and you have adverse and sometimes can be fatal side effects here. B blood you can't put A blood into B blood because they have the A antigens and they have antibodies against A blood. What do you think about AB blood people? Do they have antibodies against A or B?
No.
No, because what would happen? If they have an antibody against A, the A antigen, they'd attack their own cells. They have antibodies against B, they'd attack their own cells, and they'd be dead pretty quick. All right, so these guys, they have neither antibody A or B. And what do you think about O?
Alright, I have a question about the AB. Can you put AB in with A or no, because they have the B receptor as well?
That's a good question. So could we put AB blood into an A blood person? What do you guys think?
Like, I don't think you wouldn't be able to because it has.
Yeah, you can't, because it's still, even though it has the A antigen, it has something on it that you're going to recognize as foreign. And so the antibody B is going to attack it. All right, so absolutely. So O blood, going back to O blood, they have both antibody A and B because they have neither antigen A or antigen B. They have A and B. So, type A blood people, what kind of blood can they receive? Type A?
And O.
They can't have B because B has the B antigen they don't recognize. They can't get AB because it has that B antigen. But O doesn't have anything that they'd recognize as foreign. Even though it doesn't have the A antigen, it doesn't have anything that it will recognize and launch so therefore you can give O to A. How about B blood? You can get
B and O.
B and O. How about AB blood?
O.
O, AB. Could you give A? Yeah, you could. A blood has nothing on it that this would recognize as foreign because it has its A antigen, right? Could it get B blood?
Yeah.
Absolutely, B blood is nothing on it that this would recognize as foreign, because the L has both the A and the B. How about O blood? It can only get O blood, all right? Because you can't put any of these guys on there, in there because they don't have either one of those antigens. What's another thing of the blood type that you guys, that I haven't addressed here?
Positive and negative.
Yeah, positive and negative. Let me just talk about that real quickly here. That's called the RH factor. And this is due to another whole gene. Positive gives you another antigen. I'll represent it as a square. And negative, you don't have, it doesn't have that other antigen. So it's a third antigen on there. So, which type, O positive or O negative, can be given to anybody?
Negative.
O negative is the only one that has nothing on it, that anything else would do as form. All right, we'll stop here and I'll talk. Okay, you have a question? Okay, I just got mine.